So I'm Professor of Neuroinformatics at the School of Medicine at the University of Nottingham, and also co-director for the Center for Translational Mental Health and the Center for New Technology, Neuromodulation, and New Therapeutics. And I'm very interested in using ultrasound stimulation to improve cognition in mental health. So we're looking at depression, anxiety, schizophrenia, and other conditions. You might have heard about Nottingham before. If you know one thing about Nottingham, it's Robin Hood. If you know two things about Nottingham, the other one might be inventing MRI. So that was done here in Nottingham by Sir Peter Mansfield. And because of this history of imaging research here in Nottingham, we are hosting the National Facility for High Field MRI, which is an 11.7 Tesla a human scanner that is currently being installed and will be operational soon. So we will look at brain structure and brain connectivity and brain function with a higher spatial resolution with this facility. And we are starting the Center for New Technology, Neuromodulation, and New Therapeutics with more than 24 faculty members, particularly focusing on non-invasive technologies. There are other places worldwide looking at implants and invasive technologies, but we are very much focusing on non-invasive technology and focusing on clinical applications. So what's the demand for non-invasive, personalized, and targeted neuromodulation? We had a survey of the lay audience and patients suffering from brain and mental health conditions. We were asking them what kind of intervention they would prefer. We had almost 800 participants, around half of them healthy and half had a condition. Out of the patients, many had movement disorders such as Parkinson's disease, but we also had participants with depression, anxiety, schizophrenia, and other conditions. So when we asked them what intervention they prefer, we get this picture on the right-hand side. For the preference, we were asking them about non-invasive ultrasound stimulation, non-invasive magnetic stimulation, pharmaceutical drugs, non-invasive electrical stimulation, and then brain implants. And what you see is that there's a high preference for ultrasound. People possibly thought about pregnancy scans. So if it's safe for pregnancy scans, it should be fine as an intervention for brain and mental health. Second is magnetic stimulation. Third are pharmaceutical drugs. So people know about pharmaceutical drugs, but they also know about side effects. Then we have electrical stimulation. Maybe some people thought about electroshocks or One Flew Over the Cuckoo's Nest, so it's not very popular. But then least popular are implants. So we had many participants that said they would never have an implant categorically, so both for patients as well as for currently healthy individuals. So why was there a preference for ultrasound brain stimulation? We were asking about the perceived safety of a technique and also the perceived effectiveness of a technique. And ultrasound was seen as very safe compared to other modalities. If we look at effectiveness, ultrasound was seen as effective, but also implants, because for implants, you can directly take an electrode and put it into a target structure and stimulate that target structure. So implants were also seen as very effective. But the preference for a device is driven by the safety and not by the effectiveness. So that's the reason why overall ultrasound was the preferred intervention for brain and mental health conditions. And this article has been published a few weeks ago. So just to give you an overview of ultrasound stimulation, there's a new fast device that Brainbox is distributing. It can go quite deep into the brain, so we can reach hippocampus, thalamus, amygdala. There are several centers in the UK. So at the moment, there were 10 installed new fast systems, but there are more systems being installed and ordered at the moment. So for Nottingham, we have 2 systems at the moment, but we got NIHR funding for a third new fast system later on for clinical applications. The benefits of ultrasound are that you can be very focal. With the new fast device, it's on the order of 1 or 2 cube centimeters. If you have other devices that don't have one sound source, but 1024 sound sources, you could go down to a few cube millimeter resolution. You can directly target deep brain structures without targeting other structures that are on the pathway. So the ultrasound waves are coming out of the transducer, out of the sound source, and they're converging on one point. Only at the focal point do you have enough energy to have a change in brain activity. It's non-invasive, so we don't need implants for this focal change in brain activity. Depending on the protocol, we can increase or decrease brain activity. And devices can be used for a range of applications, could be used in humans, in rodents, or in non-human primates. And importantly, we can directly measure what the effect on the brain is while we are stimulating. So this is called an online experiment where we are observing what is happening while we're stimulating. So we can do that with MRI if you order an MR-compatible new fuzz system, or we can do that with EEG, all while we are stimulating. And just to give you some idea what might be possible in the future, for Parkinson's disease at the moment, we have systems where you have ablation of brain tissue, or you have implants for Parkinson's patients or for essential tremor patients. And this is a study by colleagues in Paris, by the group of Jean-François Aubry. And they were taking a system that is normally used for ablation, normally used to heat up the tissue and destroy brain tissue with ultrasound. So with this device, you do have the 1024 sound sources. But if you don't turn it to 11, if you only give a little bit of power, you can modulate brain activity rather than destroy brain tissue. And this is what they did in this study. So they basically had 35 seconds of sonication with ultrasound, and they then observed what happens in the patient, and they saw a 98% tremor reduction for at least 25 minutes in those essential tremor patients. It's at least 25 minutes because after 25 minutes, the surgeons went ahead and destroyed the brain tissue by turning the device onto full power. So it might last a lot longer. It might be an effect that lasts for hours or at least more minutes than the 25 minutes. But it just gives you an idea of what is possible with a focal stimulation of brain tissue. Why not invasive technology? So focused ultrasound, you can directly target a deep brain structure with a relatively high spatial precision. But why wouldn't you use an implant? So let's look at this analogy. So let's say you put a brain implant into someone, into a kid, and if you have surgery, the infection risk is 4.5%. And the device and the electrodes and the battery, they don't last forever. So if you have electrodes implanted into the brain, many electrodes will fail. So that is the case for the Neuralink system, that you have many electrodes that go into the brain tissue, but over time, many of them will stop recording properly. So you do need some replacement of devices potentially after 10 years or so, which means you need additional surgeries. If you have 5 surgeries, there's a 20% chance that at least one surgery goes bad and you do have some side effects for those brain implants. So that's not going to happen for many patients. And in particular, it's not going to happen for anyone who's not diagnosed with a condition. If you think about healthy participants who want to improve their memory or who want to improve cognition, they will not want to have implants. There's a second reason why invasive technology has a problem. So surgery has potential side effects. So ethical approval is only possible in adult patients. In some cases, there is approval for children and adolescents. But for example, Christoph Koch, director of the Allen Brain Institute, says that FDA is unlikely to ever approve implants in healthy individuals who are not patients. So the total addressable market for invasive new technology might just be 6% based on what we see for cochlear implants, where around 6% of patients with hearing loss do have a cochlear implant. So if we assume that 1 out of 3 in the general population suffer from some brain or mental health conditions, that's 2% of the total population that might benefit from a brain implant. But of course, this market will decrease further when other techniques become available. There might be digital interventions, cognitive interventions, or, as we talk about today, non-invasive interventions that could be used. Another limitation is that the currently addressed market share, so not the potential one, but the addressed one, is more on the order of 0.01% of all patients. So for example, for Parkinson's disease, in total, 1.4% of patients have an implant. And this market share, if it increases, will increase very slowly. We know from the history of brain implants that we see an 8% increase per year. And the limiting factor is not only novel devices and FDA approvals, but the limiting factor is also a lack of new surgeons and centers with brain implant expertise. So just to give you an example, in the UK, there were 12 centers that have DBS surgery expertise, 12 centers. And at the moment, we already have 10 neurofast ultrasound systems, and there are more systems rolled out. So we will soon have more centers in the UK that can perform ultrasound than centers that can perform DBS surgery. So let's look at the target effects of focused ultrasound neuromodulation. So I know some of you might already heard about ultrasound, so some of you might have seen this before, but I will try to highlight some novel papers that just came out recently to give you an overview. Ultrasound is a mechanical wave that is transmitted through the skull and through the brain tissue, and then the waves are converging on the focal point where we see changes in brain activity. And one idea how this might happen is that you have some parts of the membrane, some channels that are sensitive to mechanical pressure. So when the ultrasound wave arrives and you have sufficient pressure at the cell membrane, you see an opening of those cell membranes, you have a change in calcium, for example, and then you have some second messenger cascades and some changes that influence whether neurons will be active or not active. If you're new to ultrasound, I would highly recommend to look at this paper here by Murphy and colleagues. This is the iTrust Practical Guide to Ultrasound Stimulation. It tells you how to identify targets, how to look at targeting feasibility. So can the ultrasound wave actually reach the target? It's talking about the protocol selection and also about safety of the protocol, because you have to stay within FDA limits in terms of the mechanical pressure you apply and in terms of the temperature increase you see in your tissue. It talks about navigation, how to make sure you target the right structure, target exposure to see whether you actually have changes at your target, and target engagement here. So it's a very good starting point if you're new to the field. This is an overview published last year about studies in human participants, both in healthy participants and in patients suffering from brain or mental health conditions. So you can see which parts of the brain have been targeted so far. And the field is moving rapidly. So this is, even though it's published last year, possibly submitted 2 years ago, it's already historic because there are more and more clinical trials and more articles coming out. But from this publication, you have this overview of clinical studies. So you basically see in the middle of the circle which disease is targeted: depression, epilepsy, Alzheimer's disease, generalized anxiety disorder, schizophrenia, and others. Then you can zoom out to the next ring and you see which target structure was targeted. So for example, hippocampus. If we go for Alzheimer's disease, then you see whether it's an offline effect or an online effect that was measured. So online effect means that you're measuring during stimulation, and offline means that you measure after stimulation. And then at the outer ring, you have the modality that you're looking at. So it could be MRI, it could be PET imaging, it could be other biomarkers. So for depression, for example, we see a change in depression scores as a result of stimulation. So it's a nice overview which studies are out there at the moment and what has been done already. This is an online database that we established. You can see the article here, Kafarati and colleagues in eLife. This is a database that will continuously be updated with novel human ultrasound studies in healthy participants and in clinical populations. So feel free to add your own study to the database. At the moment, the article is based on a review of 32 human ultrasound studies in healthy participants and patients, and online and offline studies. And the idea is to look into this hypothesis. So can you actually predict what the neural effect at the target will be of ultrasound stimulation? So if you get ultrasound to the target, are you increasing or decreasing activity? Are you enhancing or suppressing activity at your target? And the hypothesis initially was that, okay, maybe it depends on the intensity, it depends on the sonication duration, on the duty cycle, and on the pulse repetition frequency. So we were looking at those papers that are out there and that include information about those properties. But the picture is not clear-cut. So what the blue line shows you is what would be expected in terms of the hypothesis. So the expectation would be that everything on top of the blue line would be enhancement of neural activity and everything below the blue line would be suppression. But if you look at those studies, you see that it's not a clear-cut distinction based on the blue line, which means that there might be other factors that play a role. It could be the total durations for how many minutes you're actually stimulating. It could depend on your target region as well. So there could be an internal property of the target region, a balance of excitatory or inhibitory neurons, or different cell types, or different layer thicknesses in your structure. Or it could be related to how your target region is connected to other parts of the brain. So it's not a clear-cut picture, but we get some more information about what we might expect from future studies based on the parameters we measure. So those were all effects directly in the target structure, but we're also interested in what effects we observe in other parts of the network, brain regions that are directly or indirectly linked to the target structure. So the idea is that we can reconstruct how the brain is connected. We can do that with diffusion tensor imaging, which is a special MRI protocol. So we can just put someone into an MRI scanner, run this special protocol for a few minutes, and we can then reconstruct fiber tracts in the human brain. What this picture shows you is basically fiber tract connectivity between brain regions. The color tells you the local direction of a fiber tract. So red means going from left to right or right to left. Green means going from the back of the brain to the front of the brain or the other direction. And blue means going from top to bottom or bottom to top. Unfortunately, DTI doesn't give us information about the directionality of a fiber. We know from animal studies where we basically dissect the brain that there were some one-way streets in the system. So we would assume that some of those fibers only go in one direction. But we can't measure that in the in vivo, in the alive human brain. So we're just assuming that all the fibers are directional as a starting point, but that's not necessarily the underlying biology. But just based on this structural connectivity of fiber tracts, we can predict IQ plus minus 3 IQ points or the age of a participant plus minus 8 years. And we can use network analysis to predict what the condition of a patient is, but also what intervention might be most suitable. So the long-term idea is to use this as a starting point to find out which part of the brain should be stimulated with focused ultrasound and what are the direct and long-term effects of focused ultrasound stimulation. Because ideally for patients, we would like to have some longer-term effects of treatment. We don't want treatment where the patient has to come back every week or every day in order to receive focused ultrasound. We would like an intervention where we have a couple of sessions of focused ultrasound and then the patient is symptom-free or has reduced symptoms for months or years on the long term. So in order to understand network effects of ultrasound brain stimulation, we worked with colleagues in Newcastle. So this is one rhesus monkey that received focused ultrasound stimulation of the left anterior hippocampus. So it's only one monkey, but this monkey had lots of stimulations. Okay, so it's 27 control stimulations, so no ultrasound, and we just measure MRI, and 25 sessions with ultrasound stimulation. And we have 250 kilohertz ultrasound frequency and those parameters here. And we then measure different properties of the fMRI signal. One is the strength of functional connectivity between all voxels and the targeted left anterior hippocampus. Functional connectivity tells you how similar the time series is in different parts of the brain. So you look at, let's say, two different voxels, and you observe the time series of those two voxels. And you can then correlate those two time series. If there was a strong positive correlation, you would say they were functionally linked. Okay. So that's a global measure of what is going on in the system. We can look at a fractional amplitude of low frequency fluctuation. So that represents spontaneous brain activity in the system. And we can use regional homogeneity, which is measuring how nearby voxels are changing over time. So to what extent is activity correlated in nearby voxels within a brain region? And what we see is that for the global measure of functional connectivity, where we just observe changes in the whole brain in terms of functional correlations, we see changes occurring 50 minutes after ultrasound stimulation. Okay. So it does take quite some time for changes to occur. It might be some slow process of long-term plasticity. Maybe some calcium changes are involved. So it does take some time before the changes occur, but they occur at about the same time for certain other brain regions in the system. If we look at local changes, it depends on the network structure that we are observing. So for some network structures, those local changes occur at the same time as the global changes. But for other structures, those local changes occur at a much longer time scale. So possibly 40 or 50 minutes after the global changes occur. And this is another indication that the effect of brain stimulation is related to the structure of different brain regions. So you can't always assume that you hit a target or you indirectly hit some other brain regions and then you have a certain effect. It will depend on the internal structure and organization of a brain region as well. We then looked at how the functional connectivity is changing over time. So this is a study together with colleagues at Oxford University, Jérôme Salé, who's now at the University of Lyon. This is data from 3 rhesus monkeys that were anesthetized. So before the study was in an awake monkey, and this is in 3 anesthetized monkeys. So the stimulation was looking at 3 different targets and then observing functional connectivity changes off target, so in other parts of the brain. If we just look at the first target here, anteromedial prefrontal cortex, we can look at 3 other brain regions that show changes over time. So you can see the 3 regions here. For the first one, the green curve, you see changes appearing early on after ultrasound stimulation. For the middle one, you see changes later on. And for the third one, you see an early change and a stronger change later on after stimulation. But the important message is that what you see really depends on when you measure. So let's say you have 3 different time windows that you're using. If you're a lab that only looks at the first time window, let's say 20 minutes after stimulation or 30 minutes after stimulation, you basically would say that the first area shows an effect and the second area doesn't show an effect. If you're in a lab that looks at a later time, let's say 40 minutes later, you might say that the first region has no effect of ultrasound and the second region has an effect. And if you're in another lab that looks 50 minutes later, you might see that neither region A or region B shows an effect of ultrasound stimulation. So the important point is that you need to look at the whole time series of what is happening after ultrasound stimulation, not just a single snapshot, not just a single time, in order to make sure you capture all the effects outside the target region that are happening in the brain. We moved on to studies in humans, and we developed a toolbox that can help you to determine what is the best entry point for focused ultrasound. So where should you put the transducer on the head in order to get most pressure, most mechanical pressure to your target? So this toolbox is taking MRI images. You basically determine what part of the brain you want to stimulate, and the system in R is then calculating what is the best entry point for each individual subject. So this toolbox is freely available at the link here, so you can download it and you can use it to plan your ultrasound experiments in human participants. We then wondered about how ultrasound can change cognitive performance. This is a study in healthy human participants. In the first session, we are just measuring structural brain scans and also resting state fMRI, so fMRI activity over time when the participant is just in the scanner and not involved in any task. So that's the baseline resting state that we're measuring, and we're then observing functional connectivity in this resting state fMRI recording. After the MRI, the participant is involved in a stop signal task. Stop signal task is just presenting a cross or a plus sign on the screen first, and then a green circle indicating that you should push a button. But while you are starting to push the button, you either have continuously the green circle, which means you just go ahead, or you have the red cross, which means you should stop your action and not push the button. So you have to inhibit your action that is already on the way. We can then measure the accuracy, so to what extent can you push or not push the button, and we can measure the reaction time, so how long does it take for you to push the button. So that's the baseline cognitive task that we are using. We then use the structural MRI scan to plan our ultrasound experiment, so deciding on the target and basically deciding on where on the skull to put the transducer. And we then have a second session where we stimulate with focused ultrasound with the new Fast Pro system. We then roll people into the MRI scanner, which is just 50 meters away from our brain stimulation lab, so we can get people in there within 5 to 10 minutes. We measure resting state fMRI and again reconstruct functional connectivity. And after the MRI session, participants are performing the stop signal task. So importantly, the stop signal task happens around 1.5 hours after ultrasound stimulation. We use K-Plan, which can be brought together with new Fast Pro to measure or basically predict how much pressure we get to the target. We use 2 targets in this study, the thalamus and the inferior frontal cortex. And we use those ultrasound parameters here, central frequency of 500 kilohertz. So that's based on the transducer that we have. It's a 500 kilohertz system, pulse duration of 20 milliseconds, repetition interval of 200 milliseconds, a duty cycle of 10%. You can see the pressure here and the total duration of 80 seconds. So 10% duty cycle means that we're only stimulating 10% of the time, 10% of those 80 seconds. So effectively, the stimulation runs for 80 seconds, but the actual stimulation is only 8 seconds, 10% of the time. Okay, so we have an 8-second stimulation where ultrasound goes to the target structure. So what do we see? We can look at other brain areas, and we can observe which other brain areas show changes in the functional connectivity pattern. And those tend to be areas that are functionally connected to the target structure. So you can look at functional connectivity between the target structure and another brain region, and if it's very strong, there's a higher likelihood that the other network region shows changes. On the left-hand side, you see the inferior frontal cortex. On the right-hand side, the thalamus as a target. So on the left-hand side, you see that lingual cortex and other brain structures show decreases in functional connectivity patterns in blue. And those decreases vary with time. Some brain regions show earlier changes and some show later changes. On the right-hand side, you see the pattern for the thalamus. And for the thalamus, you see that you have decreases of functional connectivity, but also increases of functional connectivity. At the bottom, you see the thalamus in the contralateral hemisphere. So we stimulate the right thalamus, and you see changes in the left thalamus. So this is what we see in terms of functional connectivity changes. Those are the biological changes we observe in the system. There are changes in terms of brain activity. But how do they relate to cognitive changes? If we look at cognitive changes in the stop signal task, we can measure the accuracy and the reaction time in the task. The accuracy stays the same. So whether we stimulate with ultrasound or not, and what target we are using, there is no difference in the accuracy. But there is a difference in the reaction time. So if we look at the thalamus, so before ultrasound stimulation, and post, after ultrasound stimulation, we see no change in the reaction time. However, if we stimulate the inferior frontal cortex, we do see a reduction in the reaction time after ultrasound stimulation. So what we observe on average is an 8% decrease in reaction time. So it is still the same accuracy. People don't get worse with accuracy, but they do get faster. And this can be very interesting for some applications where reaction time is important. So you can think about pilots where fast reaction time is very important if you don't want to crash your plane, where every millisecond counts. So there might be a benefit of increasing awareness and reducing reaction time for those tasks. So then the question is, can we actually predict those changes in reaction time? What you see here is the reaction time change on the x-axis, and on the y-axis you see the functional connectivity change, where we look at the functional connectivity between the target and inferior frontal cortex and another brain region, post-central gyrus. So the further we have a decrease in functional connectivity, the bigger is the decrease in reaction time. Okay. So the stronger the change in functional connectivity we see, the stronger is the change in reaction time. And we can also link the cognitive reaction time change with the physical effect of ultrasound stimulation. We can use K-plan to simulate how much pressure is actually getting to the target. And the more pressure gets to the target, the stronger is the effect on reaction time. So we can do a mediation analysis to basically show that the biological effect and the physical effect can help us to predict what reaction time change we see in individual participants. So what is a good control condition for ultrasound stimulation? Of course, one control condition is that you just turn the device off, but that can be perceived by the subject. So if you don't feel anything on the skin, or if you don't have any auditory artifacts, people might think that the device is just off. And one colleague of mine here at Nottingham, Jeong Jung, assistant professor in psychology, was thinking about a novel way, a new way to have a control condition for ultrasound. So the aim is to develop and validate a gold standard control stimulation for focused ultrasound by testing whether we can stimulate the ventricle rather than brain tissue as an optimal active control. We were using the same protocol that I was describing before, which is a theta burst focused ultrasound for 80 seconds. We have the sham ultrasound. We have a ventricle focused ultrasound. We have the active ultrasound, and we can then basically test what happens in individuals. So that was published last year in Brain Stimulation. First of all, we don't see any change on brain structures. We can look at voxel-based morphometry, and we don't see any change in the measurements here between ventricle stimulation, active stimulation, sham stimulation. In terms of any kind of side effects, we also don't see a difference between stimulating ventricle and sham stimulation. We can then look at functional changes after ventricle stimulation. So we want to be sure that when we stimulate the ventricle, we don't have any changes in brain activity as a result. So we want to be sure that nothing in between the transducer and the ventricle shows any changes, or anything else in the brain shows changes. So if we look at the whole brain analysis, so we look at all the voxels in the brain, there is no effect of ventricle stimulation. We can look at functional connectivity results, and again, we don't see significant changes of functional connectivity when we stimulate the ventricle. So ventricle can basically be a gold standard for focused ultrasound. So what you do for ventricle stimulation is that you put your transducer on the head, and in the active stimulation, you just go for the target structure. And in the ventricle stimulation, you just go deeper into the brain and you hit the ventricle rather than the target structure above. Okay, so it's basically a possibility to have a control condition where you're sure that the patient can't distinguish between the ventricle and the active stimulation, and you don't have any effects on brain activity resulting from the ventricle stimulation. So I was talking about transcranial ultrasound so far. One problem with transcranial ultrasound is that you need lots of energy to go through the skull, so it's a quite expensive procedure. But we're also thinking about avoiding the skull and targeting structures that are outside the skull. So I will talk briefly about ultrasound vagus nerve stimulation. If you have target structures outside the skull, you can use smaller devices, and you can use devices that you can basically take home. So it would be cheaper to have devices you can use at home for non-invasive vagus nerve stimulation, for example. And we are currently testing those devices here in Nottingham. We have active devices and we have sham devices. And we don't see a difference between active and sham in terms of patients or participants recognizing it. So there's perfect blinding in terms of the sham procedure. For the active device, it's on the market for around $500 or so. You can buy it online. It's marketed at the moment as a wellness device. So there was no FDA approval for certain conditions, but you can buy it as a wellness device. So this is using ultrasound stimulation. I can't see clearly there, but I can show it a bit later. So you have one spot that just goes into the ear to fixate it and into the right ear. And then next to it is a blue spot that is emitting ultrasound, 1 centimeter diameter. And that ultrasound is then activating the auricular branch of the vagus nerve around the ear and then activating the parasympathetic system. So the hardware of this is described in this IEEE paper published last year. And the effect is described in this paper here that is currently in press, so it should be out in the next couple of days. For this, there was a recruitment of anxiety patients. So those are participants with an anxiety score above 16. And 85% of the 30 participants were responding to ultrasound stimulation of the vagus nerve. So those participants were getting the ultrasound device, and they were just stimulating 5 minutes each day. They can choose by themselves when they stimulate, whether they stimulate in the morning or at night time or at lunch break. There is a hardware limit in the device, so we don't want people to fall asleep and then the device is on for 8 hours. So the device automatically turns off after around 30 minutes. So if we look at those participants, 85% show an effect. And this is the effect you see. And this is showing the effect in the whole cohort, including the 15% where there was no effect. So it's a total cohort that took part in this trial. In blue, you see the baseline scores for different measures: anxiety score, PTSD, depression, and sleep. And you see a big reduction already after 1 week of using the device in terms of the anxiety score, and then anxiety score goes down further. For some participants, the score goes down to 0, so they're completely anxiety-free after 4 weeks. So we're currently making more tests with this device, looking at the sham device as well, but we're also starting to look at heart rate variability, EEG changes, potentially in the future fMRI changes, and so on, to better understand what is actually happening and to look at other conditions. So at the moment, I'm co-supervising a PhD student who's looking at tinnitus as an application. Because the device is using, basically targeting the parasympathetic system, there could be a wide range of applications. So we know from animal studies and from other studies that stimulating the parasympathetic system can lead to better sleep. So this is something we see in healthy participants, for example. It can lead to improved heart rate. It can lead to reduced neuroinflammation, better digestion. There can be a wide range. We had one participant at a demo, and she had tinnitus. So she had reduced volume of the tinnitus after using the device for 5 minutes. But then she got up and went to the restrooms, to the loo, and she said it's the first time that she didn't have back pain when she got up. So there can be some positive side effects. There was another participant, and she had restless leg syndrome, and after 5 minutes, the leg stopped to shake for the first time. So there can be several effects of this device because the vagus nerve is involved in many different conditions. So what's the future for ultrasound? I mean, one future is more about engineering, making the devices cheaper than they are at the moment, so you have better applicability. And ideally, you want devices that you can use at home rather than in a hospital or research center setting. But the other advancement in the future will be to use closed-loop stimulation. So closed-loop means that you're not just turning on the device and stimulating, but the stimulation is based on the current state of the brain. So you can measure or infer what the current state of ongoing brain oscillations is, and then you make sure that you stimulate at the right time. We did that for a different modality, for optogenetic stimulation with implants. So we did that in rodents and rhesus monkeys back when I was at Newcastle University. And we could basically show that the timing really influences what the effect will be. So you can basically optimize your effect depending on whether you hit the right time of stimulation. For this, I recently got an MRC program grant here in Nottingham. It's a 5-year grant. We currently have an open RA position. So if you're interested, or if you know anyone who's interested, we're basically advertising for someone who's developing this closed loop approach for ultrasound stimulation and also for magnetic stimulation. So initially, in the first 2 years of the project, we will develop the technology and test it in healthy participants to optimize protocols. But we will also look at the ultrasound effect with MR spectroscopy. So MR spectroscopy is a special MRI protocol where you can look at neurotransmitters, GABA, dopamine, glutamate, and so on, in a certain target structure. So we can directly observe how ultrasound is changing neurotransmitter concentrations in certain parts of the brain. So those are the first 2 years where we observe the effect. We're looking at working memory, stop signal task, and motor evoked potential effects. And in the second stage, in the last 3 years, we will look at implementations in patients. We have a psychiatric cohort with schizophrenia, depression, and bipolar, and a tics disorder cohort, so Tourette syndrome. So we will look at cognitive changes and motor changes in those cohorts. And we will observe whether closed-loop stimulation can lead to a bigger improvement in terms of brain function in those individuals. There's more information about brain connectivity in this book here. So there are chapters about how you analyze brain connectivity, how brain connectivity is related to brain function, so how this can give you an idea what is happening in the system, how brain connectivity changes during brain development and during aging, how brain connectivity differs between different diseases, different conditions, whether it's developmental conditions or dementia or Parkinson's. And there are chapters also on the effect of learning and brain stimulation on brain connectivity. So you can find out more here. So in summary, ultrasound neuromodulation is preferred by patients and the public. Effects are predominantly time-constrained and spatially distributed in brain regions functionally connected with the directed stimulated area. Biological effects are indicative of cognitive effects that persist for at least an hour following stimulation, showing potential for cognitive improvement. So, for example, the faster reaction time that we see. And cognitive improvement can be predicted from the biological effect in terms of functional connectivity, and also from the physical effect in terms of the peak pressure change we get at the target structure. And this is a first step to predict dynamic effects, so what happens immediately after stimulation, but also what might happen on the long term, because our goal is to have long-term changes in connectivity, to have long-term changes in symptoms for patients. I would like to thank my team, postdocs, PhD students, alumni of the lab, collaborators, and funders. You can follow me on X and Blue Sky. And as mentioned earlier, we do have an open postdoc position in the lab at the moment. So thanks for your attention.